Good question.
There you go.
Alright, am I good?
Yeah.
Okay. So my paper kind of goes along with Tsukada's, and as Dr. Whittemore was saying, it talks about the mechanisms underlying the decrease in cortisol levels that Tsukada was talking about in her paper. And the title of the study was Aryl Hydrocarbon Receptor Activation and How it Impairs Cortisol Response to Stress in Rainbow Trout by Disrupting the Rate-Limiting Steps in Steroidogenesis. The three proteins that they focused on in this study were steroidogenic acute regulatory protein, which is STAR, cytochrome P450 cholesterol side chain cleavage, and 11-beta hydroxylase. And cortisol synthesis begins with stimulation of either ACTH or CRH to the intrarenal cells, which in fish is equivalent to the adrenal gland in mammals. And after it's stimulated by ACTH, cholesterol is transferred to the inner mitochondrial membrane by STAR, where it's sent to cytochrome P450 side chain cleavage, and that converts cholesterol to pregnenolone. And pregnenolone is then converted to cortisol by 11-beta hydroxylase. There's a lot of steps involved, but 11-beta hydroxylase is the final step involved in the conversion of pregnenolone to cortisol. The elevation of cortisol levels is an adaptive response, and in this study, they looked at the effects of synthetic chemicals and the response in rainbow trout. And they've said that one of the specific chemicals is polychlorinated biphenyls, and the three ways that this affects the stressor-mediated response is abnormal feedback, a decrease in sensitivity to the trophic hormones, and inhibition of steroidogenesis. The hypothesis directly from the paper was the rate-limiting steps in steroidogenesis are key targets for endocrine receptors acted by the AH receptor activation, and the two rate-limiting steps that were in the previous slide was the transfer of cholesterol to the inner mitochondrial membrane and the conversion of pregnenolone to cortisol by 11-beta hydroxylase. In the study, rainbow trout were given beta-naphtoflavone, which I'm going to abbreviate as BNF, and that is an AH receptor agonist. Resveratrol, which I'm going to abbreviate by RNF, and that is an AH receptor antagonist, or a combination of both to affect the AH receptor activation on plasma cortisol response and intrarenal corticosteroid biosynthesis. As I said before, the organism that they used was rainbow trout, and there were 8 groups of fish in 12 tanks. They were either fed feed laced with ethanol, which is just the vehicle to dissolve the chemicals and coat the feed, or they were fed feed laced with RNF or DNF, or a combination of both, and they were exposed to stress, which was it's a series of chasing and netting for 5 minutes, and levels of glucose and cortisol were taken at 0, 1, 4, and 24 hours after stress, just so they could get a temporal profile of the cortisol levels in the fish. And they assessed ACTH-stimulated increase in STAR, P450 SCC, and 11-beta hydroxylase by doing in vitro studies of head kidney cells in wells. And then the slices of the head kidney cells were stimulated with ACTH to see the response for 0.5, 1, 2, 3, and 4 hours. This is just describing the studies that they did in vitro, and they assessed the exposure of the kidney tissue to the chemicals and the response that they had to ACTH. What they did is they incubated the chemicals for the times that I gave before. They collected the supernatant and quantified the levels of cortisol and glucose, and they used that as a basal cortisol production rate with no stimulation by ACTH. And then the cells were then incubated with cortisol production rate. Cortisol and glucose levels were then measured. Cortisol was measured with radioimmunoassay, and glucose was determined colorimetrically, and that just means that the amount was measured by detecting a colored response that's formed by the interaction of the molecules of glucose in a dye. SDS-PH and Western blotting was used to determine the protein concentration, and if you remember from Cells and Phys, SDS-PH was, I believe it's negatively charged, and then they send it through a gel, and since everything is negatively charged, the proteins are separated according to molecular weight, and the largest proteins don't travel as far as smaller proteins. Real-time PCR was used to isolate the RNA from the head kidney, and it was quantified using spectrophotometry, and the cDNA was synthesized by reverse transcriptase. And they made a standard curve with the primers, which are shown in this table, and the point of that was they had different concentrations of the cDNA, I believe, that ranged from 10 to 3,000 picograms per reaction, and so they were able to form a standard curve and then compare the transcription levels to that standard curve when they carried out the experiments.
Exactly.
So they kind of just used it as a reference to compare the results. And it just explains how they measured out the cDNA. So they used 1 microliter of cDNA for every 20 microliters of the target gene, and only 0.5 microliters was used for beta-actin, which I forgot to tell you about was a housekeeping gene. It was a protein that was unaffected in all of the samples, so they just used, that was also used as a reference. And like I said before, the transcript abundance was obtained from from the standard curve, and it was compared with beta-actin, the housekeeping gene that was unchanged between treatments. And then ANOVA is used to determine significant time, treatment, and interaction effects on plasma cortisol levels. And these are the results. This was for the head kidney CYP1A1 mRNA abundance. And that was one thing that I didn't really understand. I know that CYP1A1 is a gene sequence that codes for the cytochrome P450 proteins. But
For one of them. So
one of them.
anything that has a CYP is one of the cytochrome P450
okay.
genes. And that one is a different pathway from steroidogenesis. That one's the detoxification pathway.
Oh,
So it's really, they use CYP1A1 as a marker of whether AHR was activated.
Okay.
So if you activate AHR, you should, if something's binding to AHR, CYP1A1 levels should go up.
Okay, that makes sense.
So it's a sort of marker of AHR activation.
Okay, so CYP1A1 was used as a marker, and as you can see, the liver CYP1A1 protein expression was, it was increased with exposure to BNF, and it was reverted back to control